Good morning and welcome to all. So in today we can discuss about the second part of historical perspectives. So in the last session we have discussed up to Koch's postulates, and we have discussed each and every points coming under this Koch's postulates and also the germ theory of diseases. Both the Robert Koch's work as well as the Pasteur's work helped to establish the germ theory of disease, which means that microorganisms are responsible for causing disease in plants, animals and even humans. Sorry, I had written plants, it is humans. Then describing the golden age of microbiology, that is in between 1857 to 1914. It began with the work of Louis Pasteur and Robert Koch. Louis Pasteur is also known as father of modern microbiology. He is also known as father of bacteriology. Many of the infectious agents were discovered during this period. And also another scientist whose name Charles Chamberlain, who was involved in the construction of bacterial filter, and it was made possible the discovery of viruses and their diseases using this filter, because it is very minute, so through it bacteria are unable to pass, but the virus can pass through it. Bacteria can also pass, but the smaller organisms such as viruses can also pass through it. So it led to the discovery of viruses and their respective diseases. The microbiology also contributed significantly to the development of biochemistry, that is the discovery of cell-free alcoholic fermentation by Buchner in 1897. It is also another important historical perspective which provided the key to chemical analysis of energy yielding process, because it is a fermentation. Next is about the development of culture media. So why the culture media we are using, and what is the purpose of this culture media? This culture media enable us for the isolation of pure cultures, which means which contain only one type of organisms, and those are especially important during the Koch's period. In the earlier times, gelatin was used as a solidifying agent in the culture media, but the disadvantage is mainly it melts at greater than 28 degrees Celsius itself, and some bacteria also hydrolyze those gelatin with the enzymes which are present within the bacteria, which are capable of hydrolyzing those gelatin. So it is not longer commonly used. At that time, Fanny Hesse, the wife of one of the Koch's assistants, proposed the using agar, because her mother, she also seen her mother using those agar as a solidifying agent in cooking. So she suggested this agar. And this advantage of this agar is it is not digested by most bacteria, and bacteria with their enzymes can't hydrolyze those agar, and it melts only at 100 degrees Celsius compared to the gelatin, which melts even though after 28 up to 40 degrees Celsius. And in the solidifying media, agar is used always in 2%. Then another important discovery was Richard Petrie. He is also another person, Koch's assistant. He developed the Petri dishes. The Petri dishes are even using now itself for the culture media preparation, and also in which we are pouring the culture media which we had prepared in the laboratory. Petri dishes. and the name Petri dish came in honor of the person who discovered, that is Richard Petri. Next is about the development of vaccines and ANTISERA. Vaccine, while we are in the last class, we have discussed about the vaccine and also Edward Jenner, who usually found the vaccination, who found the first vaccination against the smallpox using the cowpax lesions that we have already discussed yesterday. And this Edward Jenner in 1796 discovered that this cowpox, which is also called Vaccinia, induced the protection against human smallpox, and the term vaccination was derived from the word vacca. And the main the use of this vaccines are, it provides a protection which is called immunity. Next is about vaccination. It is mainly the method of inoculation of healthy individuals with the weakened forms of microorganism, which is called attenuated form. The attenuated form of microorganisms are those microorganisms who are still alive but their virulence is lost. It can't be virulent and its toxicity is now damaged or lost, but the microorganism is virulent. That is called attenuated microorganisms. Such microorganisms or strains are used in vaccination. That would otherwise, if the attenuation or if this virulence is not lost, what happens? It may result in disease. So the virulence is lost as a result of attenuation, and the effect is that it provides protection, which can be either active immunity or passive immunity from the disease upon later exposure. Next are the other contributions from the Pasteur. So there is an important question in this chapter: contributions of Louis Pasteur, contributions of Robert Koch, Germ Theory of Disease, Koch's postulates. These are important questions. So we can discuss here the contributions. Pasteur and Rox reported that incubating cultures longer than the normal in laboratories, this may result in attenuation. So when we are incubating those cultures for a long time, its virulence, the organism's virulence are lost. So the resulting organism results in attenuation, which means the bacteria is no longer causing any disease. While working with chicken cholera, which are caused by a bacteria Pasteurella multocida, both Pasteur and Rox noticed that animals injected with this attenuated cultures were resultantly resistant to the diseases. The next is Pasteur and Chamberlain developed other vaccines also, both Pasteur and Chamberlain. So these are also coming under the contributions of Louis Pasteur. And also apart from these vaccines, there are more other contributions also. So here both Chamberlain and Pasteur developed the vaccines such as attenuated anthrax vaccine, which is the vaccine for anthrax in which the anthrax strains are attenuated, in which their virulence is lost. How? Through the chemical and heat treatment. The chemical used here is potassium bichromate, and through the heat treatment, the virulence is now lost. Such vaccines of the anthrax against the anthrax is called attenuated anthrax vaccines. Another vaccine is attenuated rabies vaccine. That is how it can be prepared: propagating the virus in the rabbit following the injection of infected brain and spinal cord extract. So actually this rabies is a disease caused by dog bite, and it mainly affects the brain, spinal cord, etc. So when the virus infected, if we want to take this rabies virus, we are taking it from the specimen collection is from the infected brain as well as spinal cords. So those collection of inoculum of the virus. It is propagated in the rabbit, and this may result in the production of attenuated rabies vaccines. Next is about passive immunization, and the discoveries coming under this passive immunization were done by the eminent scientists. First of all, the work by Emil von Behring and Shibasaburo Kitasato, both of them, in which the antibodies are raised to inactivated diphtheria toxin by injection in different hosts. I think we have discussed this point yesterday. So they developed those toxoid which are used in the passive immunization, and it contained the antibodies which are specific for the toxins, and it provide protection from the disease when the individual is when it is injected in the non-immune individual. So it contained the antibodies. Passive immunization contained the antibodies, in which antibodies are raised to inactivated diphtheria toxin. When the diphtheria toxoid is injected, it may result in the production of antibodies in our body. Those antibodies are providing passive immunity to the person who are non-immune also. Next is how the microorganisms affect the environment. In the last slides we have studied the role of microorganisms in causing disease, and here the role of microorganisms, how they affect the environment, and it can be given through the example of fermentation and pasteurization, in which the Pasteur showed that the microbes are responsible for fermentation. And what do you mean by fermentation? It is the conversion of sugar to alcohol to make beer and wine. That is, sugar can be converted to alcohol. Here the microbial growth is also responsible for the food spoilage, and also the bacteria that use alcohol and produce acetic acid spoil wine by turning it into vinegar. So the bacteria that use alcohol and produce acetic acid spoil sometimes the wine, and it can be converted to vinegar. Sergei Vinogradsky, he was a scientist who worked with soil bacteria and discovered that they could oxidize iron, sulfur and ammonia to obtain energy. He also studied anaerobic nitrogen fixation and decomposition. Next is Martinus Beijerinck. He had isolated the aerobic soil nitrogen fixing bacteria, which is called Azotobacter and Rhizobium. These are both nitrogen fixing bacteria, and also he isolated those sulfur reducing bacteria also. Next is about Beijerinck and Vinogradsky, who pioneered the use of enrichment cultures and selective media. So they are the persons who first pioneered the use of enrichment cultures and selective media. Next is about the birth of modern chemotherapy. In this, the treatment with chemicals are also called what? Chemotherapy. Chemotherapeutic agents used to treat infectious disease can be either synthetic drugs or antibiotics. And here I am discussing other important agent, that is quinine, that are taken from the tree bark, was used for treating malaria. Actually this quinine are more prevalent in India, and that's why, and also in some countries in the last few months, certain this quinine can be used for the treating this corona sometimes. So this quinine are also used in some countries. In 910, Paul Ehrlich developed a synthetic arsenic drug which is known as salvarsan, and it can be used for treating syphilis. In 1928,